Hello students, you are welcome to Genetics series, a collection of lectures, talks on animal genetics, plant genetics, human genetics. Don't forget to subscribe to this channel so you get new recordings. At the same time, click the like button, tell a friend. Module 1 is all about the cell and cell division. So, what is the cell? Cell is the smallest unit of organisms which makes up the tissues of eukaryotes and prokaryotes. Eukaryotes are higher plants and animals, and the prokaryotes are unicellular organisms like bacteria. Basically, a cell is made up of two major parts, the nucleus and the cytoplasm. The nucleus is the control center of the cell and contains a network of materials called the chromatin. This chromatin condenses just before cell division to form the chromosome, and the chromosome is the gene carrier in plants and animals. The nucleus also contains the nucleolus. These are round bodies attached to specific chromosome regions called nucleolar organizers. The nucleus plays a vital role in genetics. This is the structure of a typical plant and animal cell. You can see that most of the organelles are similar and occur in both plants and animal cells, although there are some basic anatomical differences. For example, the vacuoles are much larger in plant cells compared to the animal cells. Also, plant cells have a rigid cell wall, unlike the animal cell, which is not so rigid. Also, in plant cells, we find organelles known as chloroplasts. The chloroplast is very important as it plays a key role in photosynthesis. The cytoplasm is the major part of the cell, and because of this, it houses a large number of organelles which perform specific roles in the cell. It is within the cytoplasm that protein synthesis takes place. In it are also found plasmids. These are special organelles that transmit some hereditary characters, and the type of heredity controlled by the plasmid is called cytoplasmic inheritance. Now, the functions of the organelles found floating within the cytoplasm are given in the next slide. So these are some of the organelles that you can find in plants and animal cells, their description and their functions. Like earlier said, the cell wall is found only in plants. It's an outer rigid layer of the cell, strong, stiff, and made of cellulose. And because of this, it gives support and offers protection to the plant. Although it is strong, it still allows water, oxygen, and carbon dioxide to pass into and out of the cell. The cell membrane is found in plants and animals. In plants, it is the inside cell wall, and in animals, it is the outer layer, made of cholesterol and selectively permeable. It supports and offers protection just like the cell wall in plants. It controls movement in and out of the cell, and it acts as a barrier between the cell and its external environment. Therefore, it helps in maintaining homeostasis. The nucleus, found in both plants and animals, is a large oval-shaped body and is the main control center as it controls all cellular activities. The nuclear membrane, also found in plants and animals, and this surrounds the nucleus, is selectively permeable. It controls movement in and out of materials in the nucleus. Cytoplasm, both found in plants and animals, is a clear, thick, jelly-like material, and the organelles are found floating within the cytoplasm. So it supports and protects the cell organelles. It houses all the organelles, and protein synthesis actually occurs within it. Endoplasmic reticulum, there are two types. We have the rough and the smooth endoplasmic reticulum. It's found in both plants and animals. These are a network of tubes or membranes, and its function carries materials through the cell, including the ribosomes. In fact, the ribosomes are found attached to this endoplasmic reticulum. The ribosome itself has small bodies that are free or attached to the endoplasmic reticulum as mentioned, and is highly involved in the process of protein synthesis. So all cells are divided into two majorly. So we have the somatic or the body cell, and then we have the sex or the reproductive cells. The somatic or body cells determine the structure of an organism and permits for growth and repair of body tissues. The sex cell, on the other hand, are for reproductive purposes only, and genes are usually transmitted via the sex cells. The chromosome. Chromosomes are thread-like structures found in the nucleus of the cell, and they are the bearers or the unit of inheritance called the genes. The genes are often represented as beads on chromosomes, and where chromosomes are located is called the locus, plural loci. Genes found on the chromosomes on the same chromosome are said to be linked to that particular chromosome. So in the cell, we find the nucleus are the chromosome, and genes are located on the chromosome. Chromosomal variation. Livestock are basically sexually reproducing eukaryotes. This means that their chromosomes, the sex and the ordinary, exist in homologous pairs. Situations, however, this situation is referred to as diploidy, which is denoted by 2n, and individuals 2n are said to be diploid. However, in nature, changes occur that leads to changes in chromosome number, and such changes could lead to fewer number or more chromosomes. So it is called monoploids in the case of one chromosome set, and the phenomenon is monoploidy, and polyploidy when you have more than one chromosome set. Polyploidy, this occurs mostly in plants that produces a lot of fruits such as banana, plantain, coffee, alfalfa, etc. Plants that show polyploidy are highly productive. This is not so much likable in animals. While polyploids occur in animals, where such occur, they may be viable, but they are invariably sterile. Why are they sterile in animals? is mostly due to defective chromosome pairing during cell division, and particularly during meiosis. So examples of chromosomal variations are shown below. So when it is N, a monoploid, and when it is more than one, polyploids. And polyploids, you have the diploid, you have the triploid, and then the tetraploids. Chromosome number differ by species, and because of this, interspecies matings will invariably lead to failure or sterility where an offspring is produced, and this is because of differences in chromosome number. So we have some examples of successful interspecies matings. Just three examples. We have the buffalo when cattle is mated to buffalo, chickney or queen hen when domestic fowl is mated to the guinea fowl, and the mole is a mating involving horse and donkey. Now, when we look at the full chromosome complement of a species, this is known as its karyotype, and this is usually determined by chromosome banding, which are then numbered according to their relative size. So here are some examples of chromosome number of some livestock species. So on the pig with 38, the turkey with 82. Chromosomes play critical roles in sex determination. The sex chromosomes and the autosomes, also known as ordinary chromosomes, are found in all animals. The sex chromosome is concerned primarily with the determination of sex in animals. So all animals have one pair of sex chromosomes and the rest are autosomes. So in the pigeon, for example, with 80 chromosomes, a pair is sex chromosome while the remaining 78 are autosomes. That is to say, a pigeon's egg has chromosome 1 to 49, then an X, while a sperm cell contains chromosome 1 to 49, then an X or a Y, and this explains why it is 80. Now this is the karyotype of the human chromosome set. We have chromosome 1 all the way to chromosome 23, and they are arranged according to their relative sizes. The sex chromosomes of all livestock, with the exception of poultry species, are usually presented as XY for males, making them to be heterogametic, and XX for females, making them to be homogametic. Now, because the males are heterogametic, they are the sex determinants. The situation is reversed in poultry species, where the sex chromosome is presented as ZW. The females are heterogametic and the males are homogametic, meaning that the females are the sex determinants in poultry species. This is shown diagrammatically here. This is in other livestock, where you see the male and the female. It is the male that determines whether the offsprings are female or they are male. This is reversed in poultry species. Female, this the male. It's the female that determines whether the chicks are male or they are female. The sex cycle is a sequence of growth, DNA replication, other growth, and cell division that all cells go through. And this is divided into two. The process of cell division, we have mitosis and then we have meiosis. Mitosis will produce two daughter cells with a diploid number, while meiosis will produce a daughter cell with a haploid number n of chromosome. So during mitosis, the daughter cells have the same number of chromosomes as the parents. And mitosis occurs mostly in somatic cells. Meiosis, on the other hand, the diploid chromosome number is only restored during reproduction following fertilization. Meiosis occurs in sex cells. So let's look at the summary of the phases of mitosis. The first stage of mitosis is what is referred to as the interphase. Now, during this stage of cell growth, there's accumulation of nutrients in the form of ATP needed for mitosis, and there's duplication of the DNA. During this phase, the cell increases in size during G1, that is growth phase 1 of interphase. And most cells, when observed during this process, is the longest part of the cell cycle. And during interphase, the chromosomes are not very visible because they are uncoiled and they are not condensed. They become condensed just before going into cell division. The next phase is prophase, and during this phase, the chromosomes are coiled and the nuclear membrane disintegrates. The nuclear membrane will disintegrate, and then the spindle cells, fibers, are also formed. Then comes metaphase. And during metaphase, we see that the chromosomes have become aligned, okay, in the center of the cell. Then anaphase. Here the chromatids, that's the individual chromosomes, separate and the chromosome number doubles. So in this case here we're having a cell with 4 chromosomes. The chromatids have separated and we're now having 8 separate chromatids. Then comes telophase. During this phase, the cell divides into 2. The chromosomes uncoiled and the nucleus is reformed, 1 nucleus for each cell. The spindle fibers then disappears and disassembles itself. You should note that telophase is different from cytokinesis, although cytokinesis has begun, running almost at the same time as telophase. This pinching that we see here is known as a cleavage furrow, and it is along this pinch that the cell will eventually divide. After telophase comes interphase 1. During this phase, the chromosomes have only 1 chromatid. Then interphase 2, the chromosomes now have 2 chromatids each. Now, what is cytokinesis? Cytokinesis is the second process involved in cell division and is the process of splitting the daughter cells apart. And there's a difference between cytokinesis and karyokinesis. Karyokinesis is the division of the nucleus, while cytokinesis is the division of the cytoplasm and allocation of Golgi bodies, plasmids, and other organelles into each of the new cell that is formed. Although some people often mistake cytokinesis to be the final part of telophase, it is actually a separate process, but it begins at about the same time as karyokinesis. So cytokinesis is technically not even a phase of mitosis, but rather a separate process necessary for completing cell division. In animal cells, the cleavage, that's a furrow containing a contractile ring, develops where the metaphase plate used to be, pinching off a separate nucleus. Now let's look at the summary for the phases of meiosis. Cells undergoing meiosis will divide two times. There is the first division, known as meiosis 1, and the second division, known as meiosis 2. Now, the phases that occur during meiosis are basically the same, and they have the same name, just as in mitosis, but the number is used to indicate the division, whether it is first or second meiotic division. So therefore, in meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. And during meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. In the first meiotic division, the number of cells is doubled, but the number of chromosomes is not doubled, resulting in half as many chromosomes per cell. The second meiotic division is basically similar to the process of mitosis. The number of chromosomes does not get reduced. So let's have a look. This is prophase 1. During prophase 1, homologous chromosomes, that is similar chromosomes, become paired, and a process known as crossing over occurs between the homologous chromosomes. So these are homologous chromosomes pairing and crossing over is occurring at this point, and you can see what it led to. In metaphase 1, the homologous paired chromosome becomes aligned at the center of the cell. This random alignment pattern is what is called independent assortment. Now, a cell with 2n equals to 6 chromosomes, like we have here, could have any of the three alignment patterns shown. The alignment following crossing over could be like this, or like this. In anaphase 1, during this phase, the chromosomes, homologous chromosomes, separate, and in telophase 1, we see that the cell is beginning to divide. This stage is however absent in some species. Now let's introduce another word, interkinesis. Now interkinesis is somewhat similar to interphase in mitosis, except that during interkinesis, DNA synthesis does not occur. Recall that in mitosis, DNA synthesis occurs during interphase. Now, interkinesis occurs just prior to the second meiotic division. So, in prophase 2, metaphase 2, following similarity with mitosis, anaphase 2, and then telophase 2. And at the end of the process, 4 different daughter cells are produced. You can see that each cell produced breaks into 2, and that is why we're having 4 daughter cells. And there are the basic differences between mitosis and meiosis. In mitosis, 2 daughter cells are produced. It is 4 in meiosis. In mitosis, the daughter cells are very much identical to the parental cells. This is not the same case in meiosis. They will be unidentical. Mitosis occurs both in asexually and sexually producing organisms, particularly in the somatic cells, but meiosis occurs only in sexually producing organisms, the sex cells particularly, leading to the production of gametes. In mitosis, only one nuclear division occurs. In meiosis, there are 2 nuclear divisions, hence we have meiosis 1 and meiosis 2. In mitosis, There is no change in the chromosome number of the mother and daughter cells. The diploid state is the default. In meiosis, changes in the chromosome number occurs because it is halved. What we have is the haploid state. In mitosis, what is called cell aneuploidy does not occur. So what is cell aneuploidy? Cell aneuploidy is the possession of chromosome number that is not an exact multiple of the haploid number of the organism in question. Aneuploidy occurs in meiosis, leading to cells that are incapable of reproducing. We'll see this in some of the future modules. In mitosis, there's no crossing over, and hence there's no exchange of genetic materials. Crossing over occurs in meiosis, hence genetic materials are passed on from one chromosome to the other. Cytokinesis occurs during telophase of mitosis. It occurs during both telophase 1 and telophase 2 of meiosis. Recall that cytokinesis is the process of cytoplasmic division. Karyokinesis occurs during the interphase phase of mitosis. This occurs during interphase 1 and not in interphase 2 of meiosis. Let us also recall that karyokinesis are changes taking place during cell division at mitosis and meiosis. Gametogenesis and fertilization. Gametogenesis is a basic biological process that leads to the production of the sex cells, that is the sperm, and the egg or the ova. During this process, diploid or haploid precursor cells undergo cell division and differentiation during the process, forming matured haploid gametes. The implication is that the sperm and the ova each carries half of the genetic makeup of the parents. And depending on the biological cycles of organisms, gametogenesis will occur either by A, meiotic division, this occurs mostly in animals, or by mitotic division, which occurs in plants mostly. In animals, gametes are produced in organs called the gonads. The process of gametogenesis in males is called spermatogenesis, leading to the production of spermatozoa, and in females it is oogenesis, leading to the production of the ovum or the egg. Both spermatogenesis and oogenesis provide a mechanism through which heritable information can be passed from parents onto their offspring. So the formation of sperm and egg cells is a complex affair occurring in a coordinated sequence of one mitotic and two meiotic divisions, cytoplasmic divisions, and cellular differentiations. Any change in any of the sequence of activity, be it biochemically or morphologically, can lead to sterility in the affected parent. All gametes are derived from what is known as primordial germ cells, which enters the gonads during the early embryonic development. And these primordial germ cells are determined early in development by the presence of cytoplasmic components called germ plasm. Once the germ cells are determined, they follow a different maturation process, and also their genetic function is also different than the remaining somatic cells of the body, and this explains why they come into full function only at sexual maturity. So, the primordial germ cells could be said to be the stem cells, which through mitosis will supply spermatogonia and oogonia. In matured animals, sperm cells are produced within the seminiferous tubules found in the testes. Around these tubules are specialized cells called spermatogonia, which are usually in the diploid state. So spermatogonia is the precursor to spermatogenesis, and the oogonia, also found in diploid state, is the precursor to oogenesis. So this is a summary of the phases of spermatogenesis and oogenesis. Starting at the diploid state, we have the primary oocytes and the primary spermatocytes. By the time they get to the haploid state, we have the secondary oocyte and the secondary spermatocytes. At the end of the process, one mature ovum is produced and three polar bodies. That is in oogenesis. While in spermatogenesis, four mature sperm cells are produced at the end of the entire process. Let us note that 1. Meiotic division does not occur unless there is fertilization of the secondary oocyte by sperm cell. 2. That only one egg is released at the time following puberty and during ovulation period of the menstrual cycle. 3. The polar bodies are devoid of cytoplasmic content. That is, they do not have those cytoplasmic contents like the nucleus, the mitochondria, and others. All these contents are transferred to this secondary oocyte. So the extraneous genetic materials are removed via the polar bodies. 4. Only one of the four haploids, that is the ovum, is actually functional. 5. Females have several million ovarian follicles, about 2 million at birth, but only about 400 mature during a lifetime. 5. All the four haploid cells, sperm cells produced are functional, and 2 to 5 trillion sperm cells are produced in a lifetime of a male animal. 7. Fusion of the spermatozoa and ova during fertilization will form a zygote, and in the zygote, the diploid stage is fully restored. So we have come to the end of our lecture on cells and cell division. Hope you have learned a lot. Before you go, click on the subscribe button, like, and introduce this channel to a friend. In our next module, we'll be looking at Mendel's laws. See you there.